بسم الله الرحمن الرحيم. Let's talk about anthrax, one of the most serious infectious diseases around the world caused by bacteria known as Bacillus anthracis. At the end of this lecture, you should know that anthrax is one of the zoonotic diseases. It's an occupational disease. You should know the basic information about the etiological agent of anthrax, including morphology, transmission, and virulence factors. Clinical types of anthrax. Anthrax is a major agent of bioterrorism. And finally, the outlines of diagnosis, prevention, and treatment of anthrax. Anthrax is primarily a zoonotic disease. That means disease acquired to humans from animals. In this case, herbivores such as cattle, sheep, and goats. Anthrax can be found naturally in the soil and commonly affect domestic and wild animals. The infected animals shed the bacteria in their secretions from nose, mouth, and anus. The bacteria remain viable in the soil so long and start producing spores. When these spores are inhaled, ingested, or come in contact with skin lesion, the spores get inside the body and can be reactivated, producing the bacterial form again, which multiply and spread out in the body, producing potent toxins and cause severe illness. Anthrax is an occupational disease. That means affect people in certain occupations who handle animal or animal products, as farmers, slaughters, and wool workers. Anthrax is widely distributed all over the world because of spores' long lifetime. But we can say, Alhamdulillah, Egypt is nearly free from anthrax. Anthrax is caused by bacteria known as Bacillus anthracis. It is large gram-positive rods arranged in chains. It's an aerobic organism. It's capable of producing spores, of course, outside the tissue. This spores is central, over, and non-bulging. Anthrax is capsulated inside the tissue. Bacillus anthracis is firstly observed under the microscope and described as communicable disease by Robert Koch. Also, Louis Pasteur, which is a French biologist, first prepared the living attenuated vaccine against Bacillus anthracis. The virulence factors. Bacillus anthracis have capsule. It's a unique capsule composed of polypeptide, while all other bacteria studied before have a polysaccharide capsule. Bacillus anthracis also have a very potent exotoxin. This toxin consists of three components. The first, protective antigen, then lethal factor, and edema factor. The first part, which is a protective antigen, binds to specific cell receptors and form pores in the cell membrane and allow the entry of other components inside the cell. Edema factor is responsible for causing edema, and the lethal factor cause various and severe manifestations of the anthrax, including killing of the cell. Clinical types of anthrax. First, cutaneous anthrax, which is the most common form. Then, pulmonary anthrax. And intestinal anthrax, which is a very rare condition. Cutaneous anthrax or malignant pustule. It develops when the spores infect skin abrasion, usually in the face, neck, and arms. It has an incubation period up to 7 days. Mostly it is not a fatal condition, but it requires proper and early treatment. It is characterized by black scar surrounded by marked edema. Pulmonary anthrax or wool sorter disease. It results from inhalation of spores in wool of sheep. Its incubation period is up to 6 weeks. It results in life-threatening pneumonia, hemorrhagic megastritis, septic shock, and death. It is a highly fatal condition. Intestinal anthrax. It is the least common form. It results from eating of contaminated meat, and its incubation period is up to 5 days, and the main manifestation is bloody diarrhea. It is a highly fatal condition. Anthrax is one of the most likely agents to be used in biological attacks. A biological attack or a bacterialism is the intentional release of viruses, bacteria, or other germs that can sicken or kill people, livestock, or crops. In case of anthrax, spreading of the spore in the atmosphere results in great outbreak. In 2001, an outbreak of 22 cases occurred in the United States of America. The outbreak was caused by spreading the spores of the organism through the mail. Spreading the spores of the anthrax in the atmosphere also can be done by the aeroplanes. Laboratory diagnosis of anthrax. First, specimen collection. You should know that this specimen from infected animal or human is highly infectious and all protective measures must be taken. The specimen is according to the site of infection. That means in case of cutaneous anthrax, you can take exudate from the skin or pus. In respiratory anthrax, you can take sputum. In intestinal anthrax, gastric aspirate or stool, and in severely ill patients, blood samples are taken. Stained film. We can demonstrate gram-positive rods arranged in short chain and capsulated. In gram strain, the capsules appear as a halo zone around the bacilli. And to demonstrate the capsule, John McFadden described a special stain to observe this capsule under the microscope by a special stain called polychromethylene blue. The capsules appear as a purplish coat around the blue bacilli, and this reaction is called McFadden reaction. Culture. On Newton to agar, it grows aerobically and shows rough colonies with irregular outlines. Rough colonies with irregular outlines, which is like Medusa head appearance. Culture on blood agar medium shows no hemolysis. Culture on gelatin medium results in slow liquefaction and giving the inverted fear tree appearance. Biochemical reaction. Cellulose and cirrhosis ferment glucose, maltose, and sucrose with the production of acid only. It's also catalase positive. Serology. ELISA test to detect the antibodies in the serum of patient against the components of the toxin. ASCOS test is one of the serological tests. It was used in the past in the diagnosis of anthrax. It is a precipitation test where the extract of infected tissue is introduced in one side of capillary tube and the specific antibody is introduced in the other side. If a line of precipitation is formed, it is positive. And finally, molecular diagnosis by PCR for rapid diagnosis, especially in case of bacterial. Control. Vaccination by a cell-free vaccine containing the PA component of the toxin for people in high-risk occupations. This vaccine is not licensed for use in children under age 18, all adults over the age of 65. Also, pregnant women are contraindicated to take this vaccine. Active immunization of animals with Pasteur vaccine, which is a living attenuated vaccine of bacillus and cirrhosis. All the infected animals are considered the main source of infection, so protective clothing and gloves for handling of these infected materials. Infected animals should be burned or buried in lime. Infected animals should be completely burned in incinerators or buried in lime. All the hair and wool and other animal products must be sterilized. Treatment of anthrax must be started early. Penicillin, doxycycline, and ciprofloxacin are drugs of choice for treatment of anthrax conditions. Thank you.